Pentose phosphate pathway is a pathway of carbohydrate metabolism in the cell which is aimed not at the production of ATP but at the production of two other types of products. One of the important products produced is 5-carbon sugar ribose, which in turn is used for the production of nucleotides including ATP and other nucleotides which are later to be part of nucleic acids. The second important product of the pathway is NADPH plus H plus, which is required for the reductive biosynthesis of lipids, steroid hormones, etc. This pathway is not meant for the production of ATP, but it involves interconversion of different sugars ranging from 3 carbon atom sugars to 7 carbon sugars. This pathway may conveniently be divided into two phases: oxidative phase and non-oxidative phase. During the oxidative phase, NADPH plus H plus is formed during two reactions. These NADPH molecules in turn are used for glutathione reduction and also reductive synthesis of lipids. And also the 5-carbon sugars, ribose, 5-phosphate, etc. are also formed during this phase, which in turn are used for the synthesis of nucleotides and coenzymes such as FMN, FAD, etc. Then nucleic acids such as DNA, RNA, etc. And during the non-oxidative phase, the ribulose 5-phosphate is reacting with a variety of other sugars of different carbon length to form glucose 6-phosphate. What's the role of NADPH plus H plus in detoxification of the radicals in the body? Hydrogen peroxide is formed as a result of normal metabolism taking place in our cells. This hydrogen peroxide will damage lipids and proteins and nucleic acids, etc., which should be prevented. Therefore, there is an enzyme glutathione peroxidase. This enzyme makes use of two molecules of glutathione GSH, thereby breaking down hydrogen peroxide into water. But in that process, these two glutathione molecules are dimerized, GSSG. To convert them back into the glutathione, NADPH plus H plus is required. This NADPH plus H plus is coming from our pentose phosphate pathway. Look at the oxidative phases. You need not study the structural formula of the intermediates. But you should know that NADPH plus H plus is formed in two stages, two reactions. First one is when the glucose 6-phosphate is converted into 6-phosphogluconolactone. The second one is when the 6-phosphogluconate is converted into ribulose 5-phosphate. In this step, carbon dioxide also is produced. That's why a 6-carbon compound is converted into a 5-carbon compound. So these are the two phases where NADPH plus H plus is produced. And also remember, ribose 5-phosphate is a 5-carbon compound which may be utilized for the production of nucleotides and then for DNA, RNA, etc. Here you can see the non-oxidative reactions, that ribose 5-phosphate is transformed into glucose 6-phosphate. These reactions are essential because oxidative reactions are possible from glucose 6-phosphate only because this glucose 6-phosphate is regenerated from ribose 5-phosphate in those cells where the 5-carbon sugars are not utilized for synthesis of, say, nucleotides, etc. Two enzymes are particularly taking part in pentose phosphate pathway. They are trans-aldolases and trans-ketolases. They are not taking part in any other pathway. What are these trans-aldolases? They are enzymes which are transferring 3 carbon fragments. You look here, CH2OH, CO and CHOH. This is a 3 carbon fragment. Whenever a 3 carbon fragment is transferred by an enzyme, it is called trans-aldolysis. While trans-ketolysis are enzymes which catalyze the transfer of 2 carbon fragments.